This is the MeatEater podcast coming at you shirtless, severely bug-bitten, and in my case, underwearless.
We hunt the MeatEater podcast.
You can't predict anything.
The MeatEater podcast is brought to you by First Light. Whether you're checking trail cams, hanging deer stands, or scouting for elk, First Light has performance apparel to support every hunter in every environment. Check it out at firstlight.com. FIRSTLITE.com.
You guys do your toes first, and then slide your heel in. There you go. Push. There you go.
Oh, there you go.
Nice and tight.
All right.
Oh man, I need some of these by my front door.
So this is.
Put your hand in there, Phil. And anything that comes in here is dead.
Yes. It doesn't.
There's no.
You don't want to.
There's no veterinary. There's no veterinarian care.
No, that's the other building over at the clinic.
So this is like, it's dead. Why is it dead?
Yes, exactly. So comes in, and this room here is big enough to back a semi in. So if you had like an elephant or something come from a zoo, like we've had giraffes before, that came in. So that's why the big crane, because obviously you don't want to lift that stuff up. So it comes through here, and then this is the incoming cooler.
And that upper door is for giraffes.
Yeah.
Yeah.
Big stuff.
Yeah, nice to meet you.
Very nice to meet you.
Hi, Corinne.
Hi, Corinne. And you got your hands full, but hi. How are you?
You're a pathologist?
I am. Yeah. So I like poultry and wildlife, but I'll do anything that comes across. It can range from, as you see, we got a bobcat in there, a turtle here, to a bald eagle, all the way out to like elk, reindeer, different things like that. Whatever somebody might have, it falls into wildlife. And then of course you have all your zoo species while you're on your regular necropsy, which can range from basically anything you could ever imagine. Naked bull rats to elephants.
Okay.
So as long as it fits on the table, it gets done. And if it doesn't fit on the table, it gets done on the floor.
What is the eagle, like how did this eagle come to be here?
So this one, I believe, is this the history on this one? We were checking some of this.
Melissa was just here.
This is one that tested high for lead.
Okay.
We get a history here that's provided, and it tells us what was happening.
Okay.
Sometimes it's really, really sparse, like just found dead, which often is the case in wildlife. Other times they have protracted histories if they've been into a wildlife health center prior to us receiving them.
Got it.
So medical management doesn't always work out, or they elect humane euthanasia because broken wings, things like that are not recoverable, and then we'll get them, and then we are looking for not only the cause of death, but anything else that might be there. In this day and age, with avian influenza, most of our, if not all of our birds are sampled for avian influenza prior to necropsy, just because that can be preceding them falling out of the sky, for example, and breaking a wing. We want to make sure that they didn't have AI first, or West Nile virus, or things like that, triple E, other infectious agents. Yes, this bird has wing injuries here. You'll see on this left wing, ulcerated skin. I mean, you can see that. These are pretty extensive wingspans on these birds. And you have trauma here. Oftentimes, this trauma can be something else is going on. The wing doesn't work right, and the bird starts pecking at it and will rip the feathers out, and it's still traumatized. We'll see that quite a bit. So yeah, these are pretty impressive. So we take measurements on these, and then all of these measurements go into the report, and then all of that is categorized forever. So if somebody wants to go back and look at whatever measurements that we take, or future measurements, we can always add more or take some away if they're no longer useful. And then that gives us baseline data across that aged bird, so then out in the field, biologists can take that data and say, well, here's an XYZ aged bird because of this measurements that we took in the field.
With eagles, they're sort of, we've been using them as indicator species for a lot of other things, just because the people pay attention to them. So eagles are more likely to be reported than, say, like a red-tailed hawk. So that's one way that we know what's going on with like avian influenza and raptors. And there's sort of a long-term effort around bald eagles and lead, which is something we've been looking at. So trying to make sure that we're testing for lead and seeing if things are changing. Like we did a big study in the Northeast looking at bald eagle populations and if lead impacted them, and saw that, despite the recovery, there was a decline in the growth rate. So it was kind of like a little bit of a slowdown because of lead.
How do you test for lead? How do you test for lead?
On the dead birds, it's with the liver. So it goes to toxicology, and they basically incinerate it, and then they can find the different metal particles in it.
Yeah.
The turtle.
Yeah, they're a little more challenging. They gotta get
What's
Yeah.
What's a turtle's path to get here?
Right.
Same sort of thing.
Same thing. Came in with a history. We're going to get all that together. But we would, to do this, we do an external examination.
What turtle is it?
This is a wood turtle, right?
It's a wood turtle.
Yeah, wood turtle.
It was found dead.
Found dead. So there's
Like no obvious anything.
right. Yeah. So a lot of times they're just found dead, right? And then you open them up, and they could have take your pick. You go to school for a long time to figure out all these different things. But it's kind of.
Here's the thing I'm not understanding necessarily. A person walks down to the edge of a pond in their backyard, and there's a turtle that's inexplicably dead.
Yeah.
They then, at some point, there has to be a well, who cares?
Right, sure.
Okay.
So what they typically will do is they'll contact the regional DEC or Department of Environmental Conservation office. They then have a whole list of things they want to look at, species for various projects, things like that.
I see, okay.
And then, if there's indications of, say, foul play, for example, like somebody was shooting animals out of season, those are ones that they might want to investigate. So that goes into a legal pipeline where it then goes and is evaluated further. The run-of-the-mill blue jay that's found dead on the ground may not be a sample that we would accept. We might say, let's just test for West Nile virus, swab on them, and then off we go. It doesn't go through the whole necropsy because it's labor-intensive, time-consuming, and all those things. But if it's a species of concern, fishers, for example, we're looking at a lot of different studies on them with rodenticides, and we also want to keep track of population. There's reproductive things that we're looking at with those. So all of those species, for example, are automatic. Just go and take them. We'll do something with them.
Yeah.
So it's opportunistic, but it's targeted in some regard to how we want to pick the cases that we want to pick.
So with fishers, for that example, with fishers, there's the things you're interested in looking, like you mentioned, rodenticides.
Yeah.
So people putting out poison for rats and mice.
Right. It goes
Somehow
up the chain,
finding
right?
its way into fishers, and so you're generally curious to see fishers that have died.
Yeah.
Got it.
Higher order birds of prey, raptors, any raptors, red tails, peregrines, things like that. We want to see because they are going to bioaccumulate up as we go. We like turtles because they bioaccumulate too because they live in water sources, right? So we can take fat from an animal if it has any, and then go back and look at all the various chemicals that might be in that water plant. So that's why these are sometimes really valuable samples that we don't know about at the time because it's just a dead animal and it might have just been trauma, but we can then utilize that saved tissue for other things.
We can take a look at a bobcat
I'd
if you
like
want
to
to
see
see the whole
bobcat,
process.
Yeah, for sure. Now this guy's bleeding all over.
Yeah, so I haven't looked at the history on this one yet. Oftentimes these are roadkills, and this is for tissue harvest, so we just want to have various baselines of what we're talking about. I'll take a look at everything while I'm in there. This one had a rabies extraction done, so you can actually look. So they take the top of the skull off here, and they pull out the hindbrain and the cerebellum, and then they're able to look through that and figure out, if it's rabies positive or negative.
But I mean, all that happens, he got hit by a car, and you're just taking the opportunity to just do an assessment on it.
Yep. So because we aren't going to go out and shoot these animals just to do diagnostics, so it's opportunistic in that regard.
Yeah.
Now we will strategically sample around hunting seasons and things like that, so that way we get deer when we want to get deer for chronic wasting disease testing and things like that.
Yeah.
But we also test for chronic wasting disease any time of the year that we get a deer.
You guys recently had a beaver that bit 5 people,
Yeah.
and that thing tested positive for rabies.
Yes.
I had never heard of rabies in beaver.
Yeah, it happens. It's a pretty nasty bite. It happens. I can only imagine. So yeah. I mean, any mammalian species theoretically, can get rabies. There are some that are highly resistant, but we treat mammalian species as potentially being rabies vectors,
Yeah.
meaning they can get rabies and then potentially expose anyone in the lab to rabies, especially when we're going to take the brain out where rabies is concentrated. So when we do that, we're in full PPE and things like that. Yeah, it does happen. Yeah, so on this, obviously they've taken the rabies sample. The exam here is typically done left side down. It's the Cornell approach that we have and we're all taught. It gives you a standard view of the organs. That way, when you do the necropsy, if something doesn't look like it normally does, then you know right away, hey, something's not quite right. If you do them on all different sides, then you tend to get less likely to notice those things right away. So if you do it the same way every time within species, it helps understand what's going on. So a lot of this blood that you'll see here might just be from when they did this and they put it back in the bag and it's refrozen and then it kind of smears along. So we'll do an external exam, we'll palpate all the limbs, make sure that the range of motion is correct. You can see that there's increased mobility here in the humerus here, so we actually have an exposed bone. So this is an open fracture there. You can actually see pieces of the bone there. So now either was it shot, was it hit by a car, we don't know until we start dissecting and look at the history. But that's something that we'll take note of, take pictures. We determine the sex of the animal. We do the exam all the way around. Look at the paws, look at the tail. These are all things that we look for. Certain viral infections can cause changes in the paw pads of animals, like distemper virus. That's one we'll sometimes see.
You'll see that with distemper?
Yeah, yeah, it's hard pad disease. So you'll actually see that change sometimes, and then that gives you a clue that, hey, this could be a distemper piece.
Oh, really?
Yeah. Yeah,
there's other hard pad disease?
Yeah, this is another term for it.
I never heard that before. I mean, I've never heard of feline distemper, but I never knew you could see it.
Yeah. So then what we do, left side down, the first incision is underneath, the reflected this way, and then you reflect this way, and you cuff. I was always taught to cuff from symphysis to symphysis, which is your pubic to your mandibular symphysis. There's one long cut that goes all the way from there to there.
Yeah.
You then reflect the ribs back, open the pericardial sac, we stop, lay everything out, and then you take a picture. And sometimes you might not notice them as you're in the middle of everything, because if they're hit by a car, things like that, but you can take that picture and then you can go back and sort of tease it out. And it sort of helps explain a lot of those things when you're like, what happened when you've done like 6 cases in one day? So we're taking notes all along the way. We get the weight, all these sorts of things that we do. And then we proceed through the entire necropsy, take the organs out, look at them individually. We often will take slices through every one of those organs.
Where we're going to do the podcast is the anatomy lab. So there's going to be like a ton of bones and skulls and stuff around us.
Species, yeah. So, questions?
No, sir. Thank you.
Thank you.
Okay, we're at Cornell University's Animal Health Diagnostic Center next to a stuffed two-headed calf with Krysten Schuler, a wildlife disease ecologist. Explain that to me. Like the ecologist part. The wildlife disease part I get.
Okay.
Wildlife disease expert I get.
Sure.
Ecologist? I don't get it.
Okay. You know what ecology is, right? So studying the linkages between things. So with wildlife diseases, trying to put all that together with like the biology, but then also thinking about communication, other social sciences like human dimensions. There's economic elements that I pull in to my research. There's political angles and things like that. So trying to put the whole synthesis together, all the way from the animal to people and rules, regulations, and how we can make that better for wildlife populations.
Got it. And explain, or I can run through it, or you can run through the scope of things you're working on.
We can run through it together, Steve.
All right. Current projects, very diverse in scope and species. You head up the New York State Interagency Working Group on Chronic Wasting Disease.
Yes.
And you're, again, we're at Cornell in Ithaca.
Yes.
New York. And New York is not yet a CWD state.
Well, New York has the distinction of being the only state that has eliminated CWD once it was found in the wild. The New York Department of Environmental Conservation actually found it first in captive deer and then in wild deer in 2005. And they responded pretty aggressively and they formed a containment area. They depopulated the two herds where they found CWD. They subsequently found 2 deer in the wild when they were doing some management activities, and in the 17 years since, we haven't detected it, knock on wood.
Man, the only state that's done that.
Yep.
I didn't even know until you just said that. I wouldn't have known that had ever happened.
Yeah, it's
I mean, I would have known that it's ever been eliminated from a state.
Yeah, I mean, it was, either good or lucky. Either way, it's helped out New York a lot where we haven't had the disease to deal with. There have been other what we call one and dones where it's been found in a single location and you go in and do management activity and don't find any subsequent cases. So there are conditions where that can happen, but it isn't the norm.
You feel it's we'll come back around and talk about this a bunch more later, but do you feel that it's inevitable that it'll come back?
Inevitable to New York?
Yeah.
Potentially, but New York...
pretty soon all 48, like eventually all 48 states will have it.
Well, even with all states having it, if we say that, there's 30 plus states that have it now, it's not everywhere.
Yeah.
And there's certain conditions that seem to support it better than others. So I think if we can do regulations that prohibit movement of prions, so you don't introduce it to new areas, and then you set up the environment where the disease isn't going to take off once it's there, it's not necessarily inevitable that it's going to be everywhere. And I think you can do a lot of stuff to prevent it from getting there faster. I think right now we're helping it along quite a bit.
Got it. And I guess it's like you mentioned, it's not everywhere. So the states almost become, I guess talking about it in the state sense is almost a little bit arbitrary, as opposed to maybe looking at it at like a county-wide.
Exactly.
Right? Like across the lower 48, that you'd look at it by county or something rather than by state, because the lines are kind of arbitrary. And a state could be surrounded by it, but it just hasn't turned up in the state.
Right.
But in often cases, it's managed at the state level. So it's in some way, it is a reflection of what a state might be doing or not doing.
Exactly. Yeah.
Okay. You do that. You, bunch of stuff here.
So
Moose health.
Yeah.
Moose health in the Adirondacks. I want to talk about that a whole bunch. Geographical epidemiology of bear mange.
Yep.
Meaning black bears,
Yep.
mange and black bears, white-tailed deer fawn survival, historical accounts of lead and bald eagles, which we looked at a bald eagle this morning in your lab, and how do you pronounce this? Blank fungus in eastern hellbenders.
Chytrid.
Okay.
Yeah.
I find I kind of got into hellbenders. I didn't know that hellbenders were a thing. I mean, I knew like big, aquatic salamanders, but I had no idea there was one as big as a hellbender.
Yeah, they seem made up, don't they?
Oh,
They
yeah.
can get up to 2 feet long.
Yeah.
And some of the names for them, like snot otter.
Old lasagna sides.
Yeah, they're really interesting animals, and they're super slimy and hard to hold on to.
So what is like real briefly, what is going on with hellbenders?
So hellbenders throughout most of their range have been extirpated or, aren't found anymore just because of things like water quality. They need really clear, clean, fast-flowing streams. So, damming rivers and things like that isn't good for them, sedimentation. And they are also being impacted by this chytrid fungus, which is a fungus that has decimated some frog populations all globally. And there's a new strain of chytrid called B-sal, which affects salamanders as well. So we've done some work here on captive hellbenders trying to see if we could vaccinate them against this fungus. So we had them here at the lab, and gave them some topical killed fungus, just like you would get in sort of a vaccine. And it really didn't do much to them. They got sick, but they didn't die. We were expecting them to die from it. And so what we've seen in working with partnerships with the New York State Department of Environmental Conservation and the Seneca Nation of Indians is that it looks like they can survive with low levels because this chytrid fungus is found in the water. And so it's kind of ubiquitous everywhere. And so, they're at low levels anyway, just because of habitat conditions, but this is just another challenge that they sort of have to deal with, but it seems like they're dealing with it okay.
And that came from somewhere new?
Chytrid, it's been introduced all over the world. Basically, the way that they used to test for pregnancy was through xenopus frogs, these African clawed frogs, and they had chytrid, and hospitals would
the way
use
they used to test for human pregnancy.
Yeah, like in people.
To me.
So I don't know the intricacies of it since I'm not, a human person, a human doctor. I am a human person. But they would use frogs and inject them to see if they would start producing eggs, and it was a way to tell if human females were pregnant. And so a lot of the hospitals that had these frog colonies to test for pregnancy released them after they, developed new tests. And so that sort of
when was that a thing?
Oh, decades ago. So
a hospital would keep a little gang of frogs around.
That's the story. I was not alive when this was all happening, but there's other species now that can harbor chytrid, like American bullfrogs can have chytrid, and then it gets in the water and other species are exposed to it. So it can do a number on native frog species.
So at some point they cut their frogs loose, their pregnancy tester frogs. This is perhaps an explanation.
Yes.
They cut the frogs loose, and these frogs had developed this.
They had the fungus, like they were used to it and fine with it. But then
introduced it into the wild.
Yeah, we see. I mean, there's introduced pathogens all the time, so we can talk more about that with some of these other things that come from different places, and then native species can't handle them.
Got it. I want to hit a correction real quick from someone we both know, Jim Heffelfinger. The other day we did we're standing. Also, where did it go? It was like a puppy display. Oh, there it is. All those puppy feet on display. We recently did an episode. What episode number was it, Krim, with Dr. Perry?
It's the...
Whatever the hell. It's called Dire Wolves and Ancient Hunting Dogs.
Mm-hmm.
We got to talking on there about taxonomic lumpers and taxonomic splitters.
Episode 466.
Episode 466. We got to talk about the taxonomy of wolves. And I use an example of what I use an example of the somewhat arbitrary divisions that will occur in wildlife. Meaning, once upon a time, you had wolves that, if you just look at the Western Hemisphere, you look at North America, let's just say North America, you had wolves that existed from the top of Alaska way down into Mexico. And there was no distinct borders between these different wolf groups. So at some point you kind of divide them up, even though no matter where you put a division line, there's like genetic crossflow. So if you'd have traveled from Alaska, northern Alaska, down into the Sierra Madre of Mexico, let's say, you would have always been by wolves. And you would gradually notice that those wolves changed, but there wouldn't have been actual lines of demarcation between these wolves. They would just get smaller and different colors, but you wouldn't have been able to really say they were all Canis lupus. You would never been able to say like, oh, these wolves are fundamentally different than the wolves a mile that way. Right? A line of continuity. I'm looking at Krysten, but this isn't her. This isn't your area.
I know, I don't know where you're going, so.
I use an example of the Mexican wolf, okay? And I was using the example of the Mexican wolf in I-40, meaning that they're saying that there's this sort of arbitrary division where if a Mexican wolf crosses I-40, it becomes a normal wolf. That was wrong. I was saying if a Mexican wolf crosses I-40 going north, he becomes a different wolf. Heffelfinger wrote in to say, frequent guest and commentator Jim Heffelfinger was saying, they're always Mexican wolves. Okay? When they cross, well, he says, here's what he says you're screwing up. When they cross I-40 going north, they leave the experimental non-essential 10J area, okay, and become a fully endangered Mexican wolf. So south of I-40, they have greater management flexibility because they're an experimental introduced population. There's just a different management structure. So that thing remains a Mexican wolf. It just enters a different management zone when it crosses I-40. He goes on to further correct. Mexican wolves are physically, genetically, and ecologically different from southern plains wolves that were in northern Arizona and northern New Mexico. He says there's no way they could have retained those differences if they freely interchanged with wolves in the north like you said they did. Mexican wolves existed in almost complete isolation in the Sierra Madre of Mexico and the Sky Islands of southern Arizona and New Mexico, with large expanses of dry desert and a lack of ungulates between them and the wolves to the north. This is why they are listed separately from other gray wolves on the ESA. People who are not, this is all a giant quote. People who are not familiar with wolf distribution historically in the Southwest often like to say that Mexican wolves just naturally blend into other wolves without a definite break. I think most of the other subspecies did blend into one another because of their adjacent distributions, but he points out that is not the case in his argument with Mexican wolves. That was a correction, Krysten.
Excellent. Good job, Jim.
Okay, let me hit you with another thing from the news.
Okie dokie.
Remember I told you this is a news item, but I'm moving into not a news item?
Okay.
We're going to talk a bunch about CWD and some other wildlife diseases. And you and I were recently at a little conference about CWD and kind of like updates about CWD. And for you listeners who are unaware, CWD is, as we said, chronic wasting disease. It's a prion disease. It's an infectious disease, right?
Yeah, you got 10 out of 10 in your quiz at the meeting. So
that's great. I don't think I told Corinne about that, my high score.
Did you eat Brody?
It's an infectious disease that attacks deer, attacks all cervids.
Not all cervids.
Sorry, what am I saying? Not all cervids?
Not all. Yeah. It's mostly white-tailed deer, mule deer, elk, moose.
Okay, what cervid has never had it?
Like fallow deer and some of the other, you can use like muntjac deer and give it to them experimentally, but like naturally infected deer, it's only those 4 species so far.
Is that a
Not
distribution?
Caribou. Caribou, yes.
Because I thought in Europe they had a caribou infection.
They do, yeah. Sorry, I was just thinking in North America.
Got it,
yeah,
okay.
reindeer in Scandinavia have...
Reindeer in Scandinavia.
Mm-hmm.
Okay. But it's so far not in any North American caribou.
No.
Herds.
They've done some experimental trials, and so there's concern about them, but they haven't been listed by the USDA yet, even though they seem like they're susceptible.
Okay. So, to date, and maybe forever, I don't know. To date, white-tailed deer, mule deer, elk, moose.
Yeah, North America.
Okay, in North America. So a news piece came out, reported from, where is this damn thing? Got it. Okay. UW-Madison researchers have found that black-legged ticks, commonly referred to as deer ticks, can harbor transmissible amounts of prions. Are you a prion or prion person?
Prion.
The protein particle that causes chronic wasting disease in white-tailed deer. What do you make of that?
Well, there's a couple different parts of it. So I guess I'm not surprised that they found prions in blood because we knew that happens already, that prions are found throughout the infected animal's body. It's just different concentrations. So the most prions are going to be in the central nervous system, like the brain, the spinal cord, in other lymphatic tissues, which is why we sample the lymph nodes, like the things in the throat there that are easiest to get at. Spleen are pretty high, eyeballs are pretty high in prions, but they're in the blood, they're in muscle tissue, they're in urine, but in much, much lower concentrations. And now we have very sensitive assays where we can detect those lower concentrations, which weren't previously available. One that I think was used in this study is called RT-QuIC, or real-time quaking-induced conversion. It's if you're familiar with PCR, which is a lot of testing that we do, it's sort of like that. So if you have one little piece of something, you can amplify it multiple times. And so, they were able to feed ticks on membranes with prion-infected blood and then look at the ticks, and there were still prions in there. And they also collected prions off of deer in the wild that were CWD infected, and they were able to find prions in that blood too. So that's another way that prions could get around, I guess. But
because it's not, but it's not totally. I guess why I bring it up is something that I should clarify my question about it. Is we can't always explain how it moves.
Right.
It'll just it'll emerge in the disease will be found in some place, and granted, you're not able to test every deer.
Right.
We're not testing every deer on the planet.
Mm-hmm. Yeah, we can get to that. Let me just finish this one thing. Where they say transmissible amounts, but that's the issue, that we don't really know what transmissible amounts of prion are yet, and we don't know that a tick feeding on something else would have the mechanism to push the prions back into whatever they're feeding on, because usually after a tick feeds, it molts and goes through another growth phase, or if it's an adult, it lays eggs and dies. So the odds of it, a nymph that would feed on a deer that has prions, then molts and goes through another stage and feeds on another deer, that it would have enough prions to push through the skin into the blood to cause an infection. I don't know that that's happening yet.
I see what you're saying.
So, sure, prions could be there, but whether that's actually going to do anything or not, we don't know.
Do you feel that when you see that kind of story, and it makes a suggestion of that, do you feel that there's a risk of it being misunderstood?
Well, I think CWD is a really complicated disease in general, and the one truth that I've kind of found with it is it always gets worse. All the news always seems to get worse. So I think it's hard to sometimes talk about these scientific discoveries because they're interesting, but as far as what that means in the grand scheme of things, and how we should take that as scaling it up to management at for a state wildlife agency is sometimes a little bit challenging. But you asked about movement of prions, and that's something that I work a lot on and I'm really interested in, because in New York, we were one of the first states to write a risk minimization plan. And it was pretty simple, one, don't bring prions into the state. 2, don't let prions be out in the environment where deer or scavengers can access them. And 3, educate the public about it. So by taking steps not to bring prions into the state, and that could be in a live deer or a dead deer, so that involves the captive cervid industry, and it involves hunters, and making sure that there's steps taken so they're not bringing prions in, and then don't let prions get out into the environment. By educating hunters about how to dispose of carcasses, if you're going out of state to hunt, what should you bring back, what's legal, what's not, how can you process your carcass and, debone it so that you're bringing back the meat, the antlers or whatever other things you want to bring back in a safe manner. Those are the type of things that I'm interested in. So when people get all wound up about birds moving prions or, coyotes or ticks, that could be a problem, but it's probably infinitesimally small compared to what people are doing, moving stuff around.
Oh, I got you. Yeah. Can you explain where we spent time this morning?
Sure. We were in the Necropsy lab of the Animal Health Diagnostic Center at Cornell, and that is a place where we bring in animal carcasses. So I deal with wildlife, so we bring in specimens that may be found by the public or the New York State Department of Environmental Conservation. Biologists want to find out why something died. They bring it in, and then we go through a whole process called a necropsy, which is the equivalent of an animal autopsy. And we take tissues for testing and examine the animal and try to figure out why it died and collect specimens for other research or archiving.
Yeah, this morning we looked at a bald eagle.
Mm-hmm.
We looked at a turtle. We looked at a bobcat. We looked at a horse.
Well, we didn't look. We looked at a horse. We didn't look at it.
Oh, so I said we looked at a horse.
Well, yeah.
We didn't examine a horse.
Examine. There you go.
There was a horse that was collaterally observed. There was a collaterally observed horse, and we partially examined a turtle.
I don't want people to think I'm looking at horses on the side. And,
No. There was a horse present.
Yes.
Other people had interest in the horse,
Mm-hmm.
and we just observed the horse being there. So I've done this before. A person, a hunter, a fisherman, a homeowner, whoever, comes out and they shoot or discover or whatever something that doesn't look right.
Yep.
It's acting weird. A coyote shows up in their yard. It's missing its hair. It's trying to bite their dog. They call their game warden.
Yep.
Whoever. They're like, something's not right with this thing. What's going on? It, by some avenue, winds up with folks like you.
Yes. So there's a whole process, and we go out and do training for the biologists in the state to tell them, what to look for, and try to educate them about different diseases. So if it is a coyote with hair missing, they are familiar with mange, and they know what to tell the homeowner. And if it's something serious that they want us to take a look at, they'll, either tell the homeowner to shoot it or they'll come out and euthanize the animal. They know how to keep it cool enough so the carcass is still in good shape to get it to us. And so there's our lab here in Ithaca, but then the New York Department of Environmental Conservation also has a wildlife health unit over by Albany, New York.
Mm-hmm.
So having multiple locations makes it so they can get carcasses, the biologists can get the carcasses to us relatively quickly so we can figure out what's going on. So they let us know what species they want to prioritize looking at, which populations, which areas are important to them, and we also let them know what diseases to look out for, what might be coming down the pike that we've heard about from other surrounding states as being problematic, and ask them to be on the lookout. So like for chronic wasting disease, we always want to hear about sick or abnormal deer.
Okay.
So those are ones that we say, yes, like if you get a call from anybody about that, go out, get that deer. We want the whole thing to come in here and take a look at it. Whereas like bear mange, we did a pretty intensive study for a few years. And we said, yes, we want to know about every report of bears with mange. And if you can get your hands on any, we want you to bring them in. We published a paper on that. We said, okay, we kind of know that we have bear mange now. We know where it's happening. But right now there's sort of a larger regional study where other researchers from the University of Georgia came to us and said, hey, we want samples from all these states. Can you help us out? And we said, okay. And there's different protocols. So if a biologist is out there, he or she can do different things, anywhere from like taking a skin scraping to bringing in the whole animal, and we'll take samples and then send them to Georgia for participation in that.
Is black bear mange killing black bears?
It does kill black bears, and Pennsylvania's been doing a lot of good work on this because when we first started with mange, we kind of said, okay, if 50% of the body is hairless and the animal looks pretty bad, you should probably euthanize it. We don't think it's going to recover. But some of the work being done now indicates that you can treat bears and they recover, but some bears, even with severe mange, seem to recover on their own. So it's kind of a crapshoot on whether they're going to recover or not. But
Well, how is it new?
it's expanded in its range. So it didn't really occur in New York until, I'll probably get the year wrong, but in the last decade or so, it's become more prevalent. And so one of the questions was, is it a specific, it's Sarcoptes scabii, but is it specific to bears, or is it the same Sarcoptes scabii that occurs on coyotes and foxes? Because we know red foxes get mange and it usually kills them. So did it jump from a species, or did it become more speciated to just being a bear one? But it looks like it spilled over from other species and just is going through bears now.
Like at some point, that happened here, or at some point it happened somewhere else?
It's probably spilled over multiple places because in New York, there's two sort of different areas of bears. So you've got the southern tier and the Catskills, which is continuous with Pennsylvania, and bears, move across the PA line. And then there's bears up towards the Adirondack. So there's sort of the northern population. And then there's a strip kind of where we are. Bears are just sort of coming into being here. They weren't there.
You're
for
seeing
a long time.
more and more of them. And that mange will hit them and they'll lose their hair. Yeah.
Yeah, and it's actually called lichenification, like a lichen, where they get really thick skin and they'll have bacterial overgrowth on their skin, and their eyes might crust over and they're super itchy, so they've just got a lot of ooze and crustiness.
Oh.
Yeah, they smell bad too.
Really?
Really bad.
How many of those come through here?
We would see a few a year, and when we did that research study, we found it was more likely to be adult females. We would get not a ton of them, but we would probably have a dozen a year, and then there would be more reports. One of the big things is that these bears with mange usually end up in bad places where they shouldn't be, so they'll be on like people's porches, or they'll be more likely to have an interaction with a human
because they're starving to death.
Yeah, they're in bad shape, and so then we get calls about them, and then they get euthanized.
How does something get mange? Like I remember when I was a kid, even or older than a kid, but high school, encountering encountering red foxes with mange, in a strong association with red foxes and mange.
Yeah.
Even in the trapping world, it would be that I got a red fox, but it had mange. And it was just taken as like, a thing that happened. They were out there, but people weren't seemingly like alarmed by it.
Yeah, I mean, red fox mange has been around for a long time, and so these mites can live off the animal for a little while, depending on the conditions and the moisture. They can last in burrows and things like that for a few days. So then another animal comes along and uses that burrow and might pick up the mites, and then the cycle starts again.
So it's like the disease is moved from animal to animal by a mite. Nothing's born with mange.
No.
God,
No, it's a parasite, so they pick it up, and then the females, burrow into the skin and lay a bunch of eggs, and those hatch out, and then they start burrowing and lay more eggs, and the infection just gets worse and worse.
And then talk through what's going on with moose right now and what you're interested in moose. Because when I read that you were looking at moose, I assumed it's what we keep hearing about with moose, which is like some tick-borne pathogen.
Winter tick.
Winter tick that's killing a lot of moose.
Mm-hmm.
But you mentioned another thing killing moose right now.
Yeah. So we have a project on moose right now. We've caught moose with a helicopter the last 2 years and put GPS collars on them. There's a student out there with camera traps. And within New York, it's a pretty small population. This is like the southernmost part of moose range. So we know, moose are challenged by warm temperatures. And then throughout most of their range, moose have problems with brainworm, or Paralaphostrongylus tenuis, which is a really long word. And that is a parasite that's carried by deer, and there's an intermediate host, which is a snail. So if moose accidentally eat that snail, they can get infected, and the parasite migrates through the spinal cord up into the brain, and sometimes it ends up in the eye, and it causes damage in moose.
Tell me the life cycle again.
So deer are infected. They poop. The larvae come out in the poop. The snail feeds on the poop, and then a moose accidentally
Like
eats
the
an infected.
larvae, it's a
parasite.
snail larvae.
No, the worm, the parasite larvae is in the deer poop.
Okay.
And then the snail comes along and likes to feed on the poop and
Got
picks
it.
the larvae actually burrow into the snail,
Okay.
and then the snail gets infected. Moose comes along, accidentally eats a snail, and then they get infected.
Because the snail's on a piece of grass or whatever.
Right. They're just cruising
That's
along.
a
munching.
convoluted path, man.
I know. Wait till you hear about the liver fluke. It's even crazier.
Hold on a minute. I just want to make sure I got it right. So, the deer that poops out the larva, how did it get infected anyway?
Same process where it accidentally ate something that was infected.
So there's enough stuff out there. There's enough stuff out there accidentally eating snails to keep this going.
Apparently. Yep.
But the parasite is not a snail.
Correct.
It's just a carrier.
It's a worm.
So what is the worm?
The worm is Paralaphostrongus tenuis. That's the
But
parasite.
what does it ever have does it remain a worm? Does it ever
Yeah.
have a bug phase?
Nope.
Okay. So it's just a worm.
Just a worm.
I got it.
Yeah. That goes into your brain.
All right. So a deer gets infected.
Mhm. And they pass it through.
Yep. Winds up in their
That's
digestive system.
Mhm.
They
Well,
poop.
Yeah. It there's a whole complicated process because it goes to their brain
Okay.
too, and then it lays eggs, and that's what comes out.
Lays eggs in their brain.
Yes, and that actually can cause some problems with deer, but usually deer are fine with it. But yeah, they lay eggs. It goes. The eggs end up in their lungs. They cough them up, and then they poop them out, and then through the poop through the snail, and a moose accidentally gets it.
But if the moose, let's say a moose ate the poop.
We need to get a diagram here.
I know,
we need
I'm
like some
tracking.
animals to okay, here's the poop, and
But if the moose ate the poop, that doesn't work.
no, it has to go through the snail.
It's just like what, like.
Yeah, I know, right? It's a
I mean,
lot.
it just seems like something that would just be so random. I mean, not random, but so isolated. The fact that this is happening enough to have population level impacts.
Yep.
Then
Well, I guess if you're a moose, you're eating a lot of stuff. So odds are you're gonna
big mouthfuls
Yeah.
of stuff. And,
right.
I've gone out to my garden, right? And I remember we have a story that floats around in our family where I used to have a little greenhouse, and I didn't wash the lettuce out of the greenhouse because I thought nothing could get on it because it's in the greenhouse.
Yep.
And there's a story where I look over and there's a ranch dressing coated worm making a break for it across my kid's plate.
Oh yeah.
So yeah, you're just eating stuff.
I stopped growing broccoli for that exact reason. Those things, like the little worms that get in your broccoli, are the same color as the broccoli. And I'm pretty sure I ate more worms than broccoli, maybe.
Yeah, and I've read that people would like to point out that like, oh, a vegan, you can't be a vegan because of vitamin B12. But someone's saying that you need such a small amount of B12 that you're going to get it through insect contamination on produce.
No,
It's just
okay.
not a problem.
Right.
So back to these moose.
Okay, back to the moose.
So we're talking about the winter.
Okay. So yeah, there's a whole bunch of parasites that moose are dealing with right now. So we just went over brainworm.
Mm-hmm.
Winter tick are one.
But I want to go back to brainworm for a minute.
Okay, you like brainworm.
Well, no, I want to just make sure I'm understanding it right. It doesn't bother white-tailed deer.
Mostly.
Okay, but mostly doesn't bother white-tailed deer, but it mostly bothers moose. Like it's hard on moose.
Yes.
Why?
Because we're not entirely sure yet. But my theory on this is that the worms get lost, and they think, they're used to migrating a certain way in a deer, and like a deer is much smaller than a moose, and so they kind of get lost along their way, and will end up in weird places like the eye. Okay. So when we have moose acting abnormally or are emaciated, we usually suspect that is brainworm associated mortality. But moose are what's called an aberrant host, And other things like llamas and alpacas also can get brainworm and die from it. And so these species that haven't adapted with this worm may have more of a reaction to it migrating through their central nervous system and their brain than white-tailed deer do. And so that reaction may cause more problems for them.
Okay, if it comes to something like this, how is the worm so picky anyways? Like, why would the worm, let's say you ate the snail.
Mm-hmm.
What is it about a human's body chemistry that would make it that the worm didn't take off? That the worm didn't go into your brain?
I don't know, because I don't study people.
Okay, but like...
Do we know that it doesn't?
Yeah, yeah.
Well, I mean, there's a bunch of worms that do, like, we don't hear about brainworm being a problem in people, but there are other ones in raccoon called Balis ascaris that's problematic, that's in raccoon poop. And so they tell people, like, cleaning out attics and stuff to make sure that you wear, a respirator or something and wear gloves, because if you inhale these eggs from raccoon poop, then you can get infected, and that one ends up in your brain and causes problems.
Oh, it does? Okay.
Yeah.
And then what's the cat scratch fever one?
Is that Bordetella? No, that's dog cough.
Toxoplasmosis.
Oh.
That's what I'm thinking of.
Toxoplasmosis, yeah. That is another one that, you get from feces, like inhaling the eggs and everything. So
and that can be a problem for people.
Yeah, that one also can end up in your brain. It's a different type of parasite, though. So it's like an intracellular one that causes a lot of problems. I did some work on sea otters, and they would get that one a lot, and they think it's from cat feces washing out into the ocean.
What?
Yeah, and and snails might be involved in that one too.
So, okay, back to brainworm for a minute now. I know we're jumping around a bunch.
That's all right.
I have heard, and I might be getting part of this wrong, I've heard that we're seeing that one of the problems for moose that are normally in the northern latitudes, okay, one of the problems for moose has been as white-tailed deer populations have expanded, habitat changes, okay, warmer winters have allowed white-tailed deer to move and encroach on areas that were historically moose.
Yes.
They've always, there's probably always been some intermingling, but there's been a more radical shift of deer moving into moose areas, and that being problematic for moose.
Yep.
This is what they're talking about, I gather.
Exactly.
The movement of these parasites into moose.
And so we'll talk about the other one that we think is affecting New York moose is liver fluke.
Okay.
So has anybody ever asked you about little livers? Like if you're eating a liver, I actually had somebody ask me this, and they're like, "Oh, what are those other little livers?" So they're kind of like leaf shaped. They might get
No, I know exactly what you're talking about. Yeah.
Yeah, so those are liver fluke, giant liver fluke, Basiloides magna.
Oh, no, I'm not talking about that.
Okay.
I'm talking about there's like lobes of a liver. Uh-huh. There's like a little sidecar liver on there.
Sure, yeah. Those
okay, that's what I thought you were asking about.
No, but these are parasites that are in there that some people have fry up and eat.
Oh.
Hold on, back up on that.
Yeah.
Tell me again what it looks like.
It's like yay big, and it kind of looks like a leaf. I'm sure I can find a picture.
And it grows on a liver.
Yeah, it grows in the liver. So those migrate through the liver and make these cysts that are full of like brown gunk, and they destroy the liver. And so that's the big problem.
And
that
people
we're seeing.
like eating them, or they're accidentally eating them.
They probably accidentally eat them. But I was just curious because you get a lot of weird questions.
And he thought it was the sidecar of the liver.
No, the little sidecar thing I'm talking about.
Right, I know what you're talking about. Yeah. No, these are parasites within the liver that kind of look like a liver almost, I guess. But yeah, they're different.
I want to ask you later about the nasal bots.
Oh yeah, those are fun.
Are you into those?
Why not?
Okay, but anyway, let's go on.
Okay, we'll keep going.
On to liver fluke.
All right, so liver flukes also are carried by white-tailed deer and have a snail intermediate host, but these have an aquatic snail as the intermediate host, so it's even sort of more complicated. The moose have to get this aquatic vegetation where the the fluke actually insists on the vegetation and stays there, and then the moose comes along and eats it and gets infected.
Okay, so he's got to be underwater.
Well, he can, eating in a wetland or something, eating some plants out of there and gets it that way. And so out of the moose that we have on the air right now, I think we've had...
Explain what that means.
With GPS collars on.
Okay.
That we've had about 3 already that have died from liver fluke, and these were captured as yearlings. And so we're worried about the survival of juvenile moose in the Adirondacks. So there's been a lot of studies by folks here at Cornell in the USGS co-op unit at the SUNY School, Environmental Science and Forestry, doing moose work, to see if the habitat's appropriate, to look at adult reproduction. A few years ago, we collared a bunch of females, and all the signs pointed to juveniles as their survivorship wasn't very good. So that's why we're doing this study. And so the fact that we've already seen 3 die over the course of our 2 years already.
How many moose do you have out there with collars on them?
I forget. I want to say we caught like 15 the first year and 19 the second year.
And 3 have died from that. How many have died from other stuff?
Jen, my PhD student, would have the numbers, but I don't think too many others. I think most of them are still in the air.
So you got a mortality signal. You go out there, you get it.
Yep.
You dissect it, bring it here, whatever. Dissect it, open it up, liver fluke.
Yep.
The liver's destroyed.
Destroyed.
I was actually so much that someone would know when they're looking at it.
You have to kind of get a feel for what normal is, and when you have a liver that just looks like pulp. And the other thing, I was at one of the moose necropsies, and it was full of fluid. So like its liver was failing, and it couldn't process anything anymore. So it was just filling up with fluid. Like we punched a hole in it and had a 5-gallon bucket under the table, and it was overflowing. It had so much fluid in it.
5 gallons.
Over 5 gallons. Yeah, there was so much pressure in its abdomen, like the fluid was like shooting out a couple feet when we punched a hole in it.
Oh. How many flukes does it take to do that?
I don't know, but it was a lot. Like it was pretty trashed when you got to the liver, and for an animal that young to have that much damage.
When you get to something like that, how do you ever make the determination that this is new, this has always been here, but we hadn't noticed it? Do you know what I mean? Like,
right.
How do you ever go down that path?
Yeah, it's hard because you're just comparing it to what you know already. And that's why we have challenges sometimes with saying things are a problem, because we get so many things opportunistically where we can't go out and just sample the population like we'd like to randomly and say, okay, is this normal or not normal? So yeah, we have to do some creative statistics and just doing this work over time. You get a baseline, so you kind of know, are things, we've never seen this before in the, New York has a really long history of doing wildlife health and knowing what's normal and what's not. So those people are really good resource of institutional knowledge to compare against, to say, hey, what do you normally see?
And to return that real quick to the work you guys are doing downstairs we were looking at this morning, is that gives you that influx of animals coming in,
right?
Gives you a chance to look for this stuff,
right?
Meaning someone hits a moose with a car, the moose comes in, and you'd be like, oh, does not or does,
yeah,
right? And it allows you to start getting a snapshot of what's going on out there, even though, like, we looked at a bobcat this morning that obviously, I shouldn't say,
that one was
fairly obviously, like, probably from the report of whoever brought it in, but also just like at a glance, it's like, this thing's been run over by a car, bad. But you're able to check to see what its health might have been like prior to it getting hit by a car.
Exactly. Was there something that may have predisposed it to getting hit by a car? So it's sort of a judgment call. Like, we don't run every test on every animal because that would be prohibitively expensive. But by saving the tissues, if there's sort of a potential smoking gun out there that might have caused something, like, for some of these animals, like maybe they had something, on board, or maybe they were sick and that's why they walked in front of the car. Maybe they weren't quite right. So,
Yep.
Yeah,
Krysten was saying to me earlier, you were talking about found dead.
Mm-hmm.
And you were saying, tell me more. Found dead.
Found dead isn't really helpful. Like, we know the animal's dead already. Like,
found dead below a power line.
Yep.
Found dead in a bird's nest.
Mm-hmm.
Found dead below a big window.
Right.
Found dead where?
Yeah.
Found dead after it bit your dog.
Exactly. Right. Yeah. So one of the things when I go out and talk to the biologists about sort of doing like a crime scene investigation type work, because a lot of times they get tunnel vision on the animal and they go and pick up the animal and they're worried about putting their gloves on and doing everything right. But, letting us know what the situation is, because that is really a huge part of solving the puzzle. Is it hot out? Is it, is a species migrating? Is it, we've been seeing sort of low level mortality in this area for a long time, or, we found 20 turkeys dead and there was a smoking tree because it got hit by lightning. Those types of things are really very helpful in getting a diagnosis, because a lot of times carcasses will come in and, if it's found dead, you don't know how long this thing's been dead for. And if it's hot out, they tend to rot really quickly. And when that happens, the pathologist will call that autolyzed. And so, it's different than having a pet that might be euthanized at a clinic and then you open it up and it's, fresh.
Yeah.
These things, a lot of times we don't know how long they've been dead for, and they rot pretty quickly depending on the conditions.
When earlier we explored the idea of being a disease ecologist, and you talked about, because you're looking at a complex situation with all these other inputs.
Mm-hmm.
If you're looking at a lot of moose and you're seeing that in northern New York, you're having a high prevalence of liver flukes in moose, you collar moose, a significant percentage of those moose die from liver flukes. You're not the one that declares that liver flukes are having a population level impact on moose, or are you? Or does that information then go to down the road to someone else who's doing population level work?
Oh no, we work really closely with the New York Department of Environmental Conservation.
Uh-huh.
And so we have a PhD student working on this, because it's a research project with them to try to define what's happening with the moose population. So yeah, the student will do population modeling, will help out with that based on, the mortality information that we have from, collected over the years. She just sent me a manuscript about it. We'll try to figure out what's going on with that population, and then we'll feed that directly to the state agency so they can put it into their moose management plan and decide how they want to proceed.
Had it been flagged prior that moose numbers are declining, and that opened up a sort of investigation about what is going on?
Exactly. So there was work done by a lot of other researchers looking at archery hunters' sighting logs to try to estimate the population. There's been helicopter surveys for moose, trying to estimate the population over time and see if it's, stable, declining, increasing. So, and then like all the mortality tracking that we've done and trying to determine cause of death. We did some preliminary modeling to say, okay, we think it's juvenile moose that are being most impacted. So those are the ones we should emphasize for putting the collars on. There was a previous helicopter capture with adult females. And with that, we were able to work with other researchers to develop a test to look for brainworm.
Mm-hmm.
And when we tested those adult females, it showed that they had exposure to brainworm, but they obviously survived. So that kind of changed the dogma that brainworm might be a death sentence for moose, because we think that these females got infected and they were able to survive the infection. So yeah, trying to get a sense of that. We knew the females were also pregnant. The state biologists walked in on a lot of these to actually get eyes on calves to see if they had, singles or twins to kind of get those reproductive numbers as well.
Yep. Let's talk about the eagle for a minute.
Oh, wait, we didn't finish winter tick, though.
Oh, no, okay. Let's go back to winter tick.
Okay, so winter tick is a problem that they're seeing in Maine and New Hampshire and Vermont pretty significantly, also killing juvenile moose. And they're trying some new management strategies to decrease the population density of moose to see if that slows down the transmission of winter tick, because winter tick are another thing that deer carry that deer can manage. They can groom themselves enough to get the winter ticks off, but moose don't seem to be able to do that quite as well. So when they
they're trying to lower moose numbers,
to stop,
to save moose. That's a logic that is going to be logic that's going to be challenged by a lot of moose hunters.
Yeah. Well, I guess if you're a moose hunter right now, it's good for you, right? Like if you're out there helping lower the density.
That's how they're doing the lowering of density. But the old line, I don't know if it came from Vietnam, but the concept of burning a village to save it, right? That's hard for people.
Oh, I know. I deal with that all the time with chronic wasting disease.
Yeah.
But in New York, we hadn't seen winter tick the same way that other Northeast states had until recently. So with some of the captured moose, when they do assessments for winter ticks, they look for them on live animals, and they found more than we had seen previously. And we're starting to get some game cam pictures in that looks like the typical hair loss pattern that you see on moose, where they're missing hair on the shoulder. And some of the states that have it pretty significantly, like Maine and Minnesota, they actually call them ghost moose, where they've lost so much hair that they appear more white because they've rubbed off all those guard hairs and everything.
Oh, really?
Yeah.
Let's say you knew, not that you don't know. Let's say we know that has a population level impact, or the brainworm, for instance, or either one of these. Like, there's nothing. I mean, is there? Is there any control mechanism that would wind up being helpful?
Well, if you didn't have as many white-tailed deer in moose territory, that would be helpful.
Yeah.
So, I mean, Maine is trying really hard to talk about chronic wasting disease and deer numbers and moose, because a lot of the towns there have started feeding programs, and those are really hard to stop once they get started, because then you have too many deer, and then people obviously don't want to see them starving in winter, so they keep feeding, and so then you just can't get people to stop it because they don't want the animals to die.
In Alaska, in eastern Alaska, they have like basically a shoot on site policy for mule deer coming into Alaska.
Yeah.
Not welcome.
Right.
Around disease issues, right?
Yep.
Being that those deer are getting there due to human-induced habitat changes.
Yep.
Following road systems, right?
Yeah. You see that out west with whitetails.
Edge habitats, right?
Following rivers up.
Yeah, and it's changed. The policy on it's changed mildly, but basically it's like, if you see it, kill it.
Yeah.
Like not wanting them in.
I know it's challenging because we've, I think people forget that we've altered habitats so significantly and have an impact, and that we're not separate from nature. And I hunt deer and eat a lot of venison, but in a lot of places there's deer densities that are way too high, and they're problematic for a lot of different reasons, chronic wasting disease being one of them. So if there are ways to lower deer densities or not encourage them to encroach in new areas, that's something that I think hunters should be behind.
Can we talk about eagles?
Yes, go ahead.
Okay, you guys had an eagle.
We had an eagle.
Your interest, we were looking at an eagle. They hadn't begun the necropsy on it yet. I asked I could see if eagles were still an ESA species, Endangered Species Act protected species. I could see that, like, every eagle that died, you'd want to take a gander at it.
That's how it was for years and years.
That's not the case anymore. Eagles are recovered. Eagle populations are increasing. Why are you guys interested in eagles? And then I want to get to the turtle.
Okay.
We'll talk eagles. What's of interest about eagles?
So eagles are good indicator species for a lot of things because the public pays attention to them. If people find a bald eagle, they feel compelled to report it. So
they feel compelled to tell you that they're seeing one.
Mm-hmm,
when I have friends come up to my place in Alaska, I have to wait a few days for them to stop reporting to me
that there's eagles.
Uh-huh.
Oh, I've got a good eagle in Alaska story for you
later.
later.
You know what? Wait a minute, and there'll be 20 in a tree. And at some point on this trip, you're going to stop observing them.
Yeah.
They're just there.
I had, in my previous job with the U.S. Geological Survey at the National Wildlife Health Center, I would hear about these mass mortality events and do investigations on them. And one of them was about all these bald eagles in Alaska, where if a truck leaves a fish processing facility, it needs to be covered, because if it's not, the eagles will start dive bombing it to get all of the fish parts. And so one left and was not covered. Eagles started going into it and just kept piling in on top of each other, and 20 bald eagles died in this fish truck, just smothering each other, trying to get to the fish.
What happens after a soccer tournament?
Yeah.
Yeah.
So
like a stampede. Really?
Yeah. So a bunch of eagles died just smashing each other trying to get to the fish parts.
God, it's so un-eagle-like.
Everybody has these ideas of eagles being so majestic.
Yeah.
And, I mean, we get
Ben
eagles
Franklin didn't like them.
Yeah, he wanted the turkey, right?
Well, that's a myth, but he was being he was very disappointed in the eagle.
No. Well, yeah.
And when he threw the turkey thing, he was disappointed because the eagles are scavengers, and didn't think it was a great symbol for the country. And the way most historians regard his comment about the wild turkeys, he was saying, basically was like, puh, might as well do the wild turkey.
Ah, okay.
At least he's vain. Right.
Yeah.
It wasn't, it's much cited.
Yeah.
But most people are
they don't get the attitude with it.
Yeah, because he didn't write down puh. But there's
good that you know that.
There's an implied just make it the turkey. At least he's vain.
Mm-hmm.
But it wasn't. They think it was in jest. The suggestion was in jest.
Yeah, I see.
But he was legitimately not happy with the eagle.
Okay. Well,
a scavenger bird.
Right. And we see conspecific trauma all the time, like eagles puncturing each other from fighting and things like that. So yeah.
Okay.
I mean,
but they're a good indicator species.
Right.
Because people take notice of them.
Exactly.
And I did, man. If I was walking down the road and I saw a dead eagle,
you'd do something.
I would. Let's say I was walking down the road, and on the right-hand side of the road is a dead eagle, and the left-hand side of the road is a dead buzzard. I'm going to pay way more attention to the dead eagle.
I know. Buzzards don't get any love.
To the point where I might even notify someone about
it.
Because I would think, is someone shooting eagles?
Exactly.
Are they pissed at eagles?
People
what happened to that
them.
eagle?
Yeah. So we're looking at them for a couple different reasons. One of them is just to keep that historic trend going, where we want to keep an eye on what's happening with them. I mentioned we just finished a study a few years ago looking at bald eagles and lead.
Okay.
And so we gathered data from 7 different Northeast states, and it was diagnostic labs, rehabbers, state agencies, and put all that together to answer that exact question. Does lead have a population level impact on eagles? And so we put that together and then did some very fancy mathematics with my research associate, Brenda Hanley, and figured out that lead, we all know eagles have recovered, there's no debate about that.
Yep.
But the long-term growth rate of eagles was depressed by 3 to 5% by lead poisoning. And so it wasn't that it stopped the recovery. It was just like driving a car forward with your foot on the brake. So it just kind of, didn't make it optimal.
Yep.
And so we used a bunch of
slowing recovery.
Yeah. So it still happened, but it would have been more efficient if eagles weren't getting lead poisoning.
Yeah.
So
now, would there be? We were talking about trigger warnings.
Mm-hmm.
I'm going to issue my first ever trigger warning.
Really?
Yeah.
Wow. Do I get a plaque or something?
No, you're not going to get triggered.
Okay.
But this is I was trying to seek clarification about what, I heard it all the time. I never knew what the hell it meant. And it would be you'd notify someone if they were going to hear something upsetting.
Oh, yeah.
People get riled up about this subject.
Oh, I know. 'Cause
Yes. When we're talking about lead in birds,
mm-hmm.
We're talking about bullets.
I know.
Okay. So for you people that are going to get all one way, like no matter what, you're going to, if you hate lead ammo, if you love lead ammo, here's a trigger warning.
Okay.
Because you're going to hear something that's going to
something. Yeah, I know. Right.
And like, there's very few people who just want to discuss things. Not very few. There's a lot of people who just don't even want to talk about shit.
Oh, I know. Yeah, I know. My dad, not interested in discussing this whatsoever.
They'd rather just be that I don't want to talk about it.
Mm-hmm.
But we're going to talk about it.
Okay.
So, and I'll do my best to throw in. I'll do my best to interject, like, well, what about isms?
Okay.
Okay. We're talking about bullet lead.
Yes.
And birds get bullet lead by eating shit that's been shot.
With a bullet.
With a bullet.
Mm-hmm.
So you get a deer. Say you shoot a deer, you hit it in the shoulder blade. You're using some kind of, jacketed bullet, some kind of bonded bullet, whatever. It's probably copper sheathed around a small, what would strike you as a very small amount of lead. Okay. So most bullets there's a lot of different bullet construction, but if we're going to divide two big game bullets into two classifications for our purposes here, we would talk about like monolithic, meaning that they're all copper, or there's some combination of copper and lead. There was a time when it was just lead, but most bullets are jacketed. So you shoot a deer, you hit it in the shoulder blade, that bullet breaks up, sends little bits of stuff here and there, deer dies, everybody's happy.
Except for the deer.
Yeah,
but,
deer's dead, everybody else is happy. You got the deer. Maybe while you're gutting the deer, you have some damage, some severe damage on the opposing shoulder. You cut that out and don't even bring that home. Later, you butcher your deer at home. There's some parts that are shot up that you don't want. You take those and you go out and put them out back because you like to watch birds and whatnot, whatever. You dump it in a ditch somewhere, whatever you do. And it winds up being introduced into the food chain in the wild. Things consume it, and they can get lead. And if you're, like we mentioned earlier, a bald eagle whose specialty is scavenging,
right?
And you're a long-lived bird, and you get tuned into deer gut piles, and you're like most things where you realize that a great place to, there's certain great places to hang out in the fall because that's going on. I remember, to give an example of how would animals ever tune into this, I remember hearing about this project where there's a particular highway in northern Montana where there's really high whitetail deer populations, and it would actually impact golden eagle distribution. That there were eagles who would spend their winter, they were roadkill specialists.
Yeah, I believe it. Yeah.
They would spend their winter around 93.
Mm-hmm. Yeah.
It's like you could make a living all winter flying up and down and picking off, eat one carcass, go to the next carcass. Yes. So you're an eagle, you live a long time. Over the course of your lifetime, you eat enough little tidbits of lead that you get a lead poisoning. You get a fatal amount of lead. You're shaking your head, no?
Well, potentially, yeah. The way we looked at it was, so there would be eagles that didn't have any lead on board. Then there would be exposed eagles that had, a little bit. And we have problems. Sometimes people talk about like chronic and acute, which you're kind of talking about like chronic over the course of your lifetime. And then there's sort of levels that are enough to kill you over.
In a dose.
Yeah.
I see. Okay.
And then that would be like lead poisoning. And so people would often term, like if you get a big helping, like if you eat a lead sinker or something, that would be acute lead poisoning, and that would be a level enough to kill you.
I see. So dying from a single meal.
Yeah, we can't tell that the way that we get the data. So we don't know if they accumulate enough lead over time to kill, to get to that level, or if they just eat, happen to get a fragment that's big enough. Because a lead fragment as small as a grain of rice is enough to kill an eagle because they have a very acidic digestive system.
Okay.
And so they break that down a lot more than like people eating lead. It tends to pass through and not get as digested as it might in an eagle.
See, that's the thing I'll hear from people is they'll say that lead, and a lot of things, that lead just passes through and you're not breaking it down.
But we're doing a study now looking at fisher, so mammalian scavengers, to try to figure out, are other animals getting lead from what we term attractive nuisances, like a gut pile. And I want to say it's like 70% of the fishers we've tested from trapped animals had some levels of lead in them. So a lot of fishers are getting lead too.
Some level of lead in them in what way?
From scavenging.
No, but detected where in them?
In their liver.
Okay.
So we asked trappers to give us carcasses. We took out the livers and tested them.
And you incinerate the liver. Mm-hmm. And then it leaves behind these metal residues.
Yep.
And you can find the lead.
Yep.
And how much lead might you get out of a fisher's liver?
They were like just trace amounts, so
okay.
not super high, not enough to kill them, obviously, because they were trapped animals.
And I'm not trying to like create like a suspicion of doubt, but what else might they have done to get that lead? Is there any other?
People throw out ideas like tire weights. If there's tire weights that have been thrown off tires alongside the road.
Fishers are chewing on that?
Potentially. Old discarded batteries.
And they're chewing on that?
I don't.
But these are just things that people to create a suspicion or to throw out some possible other explanation.
Exactly. And so for some of the eagle stuff, we did try to look at the isotopes, the lead isotopes, to figure out, is it coming from bullets or is it coming from lead paint chips or whatever. And so we did that analysis. But apparently, because there's such a limited amount of lead smelting anymore, that a lot of the lead is recycled. So you can't tell where that lead actually came from.
Yeah.
So we weren't able to identify it, but
for bullet making, that's all recycled lead.
Right.
So it's all from batteries.
Yeah, we did try, but wasn't helpful.
Here's the thing people say, and this isn't your area, but this is a thing people would point to. Here you have that. Let's take the eagle example, and the eagle we looked at today will be tested for lead.
Yes.
Okay. There's an argument that it's not an argument you can ignore really, because it's a sound argument. It'd be eagles were endangered, and they were endangered because of DDT.
Right.
Okay. They were endangered because of a pesticide that would cause their shells to be very thin, and they would kill their own shells. They'd break their own eggs when they incubated them. That's simplified, but that's what happened with eagles. The use of DDT was prohibited. Eagle numbers started to increase. They were one of these few, I think I don't know what it is, 2% of the species that go on the endangered species list come off because of recovery. It's not very common.
Right.
Some come off because they're gone. They go extinct. Some come off because we realize that they didn't warrant being on there anyway. But it's a rare thing to hit recovery, and it's always celebrated to hit recovery. Peregrine falcon, bald eagle actually hit recovery. And now their numbers are growing. And someone would point to and say, well, bullet lead, yes, they're growing, but bullet lead is slowing the growth. Mm-hmm. And an argument, and it's true, would be there are many kinds of human-caused mortality that are slowing recovery. Vehicles, like they're increasing, yes. Would they be increasing faster without vehicles? Sure. Would they be increasing faster without power lines? Would they be increasing faster without X, Y, and Z? So there are many human-caused things that are slowing the increase, but the increase is there, and they would argue this is an acceptable level. This is how the argument goes. It's a like I said, can't be ignored. We as a species have an accepted level of human mortality, meaning that's like they're growing. There's a bunch of things you could do to make them grow faster, but we're not going to do all those things.
Sure.
Because there's an inconvenience level.
Well, I think that's the rub right there, is that if we're talking about lead from bullets, and hunters are hunting because, they're conservationists, they love the animals, to think about that you're causing, if you think about all the rules for shooting and know your target, what's beyond, and one bullet, one kill, all those types of things.
Yeah.
If you're leaving something out in the environment that's killing other animals that you didn't intend, to know that that might be happening, I think is something that hunters should know about. And, I'm not sitting here saying don't use lead bullets, but knowing that this is happening to other species. And if people want to make a change in how they hunt, maybe they don't leave the gut pile out in the field. If you want to shoot lead, okay, but don't leave that out there for scavengers. If you want to change your ammo, that's okay too. But just knowing that it's a problem, and getting back to your argument, like there's a lot of things out there that could slow the population growth. That's absolutely true. This is one where hunters are making this choice. And the thing that I thought was most interesting about the work we did with the population modeling, people are like, 3 to 5%, we can, okay, it's not, stop the recovery. That's not a huge number. But one of the elements that we were able to look at was the population resilience. So how much flexibility does that population have to absorb other threats and keep operating? And bald eagles are kind of maxed out. So they've stretched their population and how much they can adjust their population parameters to make that recovery happen. And so in the paper, we talked about how if other threats come along, with climate change, we're talking about using more wind energy. And if that, the wind turbines are knocking down eagles and killing them, or other infectious disease. And we actually put this paper out before highly pathogenic avian influenza came out. And then poof, that happened. And we saw a lot of bald eagles dying from that. So it's not that they can't absorb that stress, it's that you might be stressing them too much. So the same thing you were talking about with DDT, like maybe having lead and DDT was just too much for the eagles, and that's why the population was crashing. So you can't just say it was just DDT that was doing that. And you also had the ban on lead ammo in waterfowl in 1991. And so bald eagles eat a lot of ducks and, wounded animals. And so we might have actually seen less lead poisoning recently, since that ban, because they weren't eating, shot waterfowl. Now there's other places that are expanding use of bullets in different areas. Like in New York, they're adding new counties that can use rifles instead of shotguns. Okay. So it's not necessarily things like shots, like slugs that are the problem.
Slugs a hell of a lot more lead.
Right, exactly.
Like a lot more lead.
Yeah, so hopefully the eagle's smart enough that it doesn't eat that, but it's those small fragments when a bullet shatters, that go everywhere, like up to 30 centimeters away from the wound channel, that can be problematic. So
I understand I understand the point you're making, that 3% from this, some percent for that. Years ago, I was looking at and had a conversation with someone about impacts of wolves on elk. Okay. And, you start with this thing where, let's say you look and you have 100 calves, right? And you were of those 100 calves, you were always losing for many decades in the past. You were always losing 30 of those calves to mountain lions. And that stayed the same. And wolves come in. Wolves are reintroduced into an area, and you see a real decline in elk. but the wolves aren't even eating as much. They're not killing as many calves as mountain lions are. But that mountain lion thing had always been there.
Right.
They were always carrying off 30%.
Mm-hmm. Yeah, additive mortality.
There was some sort of equilibrium there.
Mm-hmm.
And you're used to that. But then you add another 7%.
And it's enough to tip it to a different direction.
And then you notice this thing, but you still look, and everyone would be like, wolves are killing all the elk. And in some areas, mountain lions are killing all the elk. The wolf thing's just a new additive thing that has disrupted what we've accepted to be normal.
Exactly.
But if you want to talk about when an elk calf hits the ground, its biggest risk remains a mountain lion. But that's just always been so consistent.
Mm-hmm.
And this is a new part.
Yep.
So I get the point that, I think that you can, that someone could make the decision, and I'll just say, like, I use copper ammunition. I use ammunition with lead in it still, all right? Like, I'm pretty well versed on the pros and cons of it, the efficacy of it. The technology on copper ammo has come a long way, but there's some, when you get into efficacy, there's some definite arguments for, there's some real arguments for lead ammunition. I have found generally that in cases where people are given, in cases where people are offered an alternative, they'll often go for it. Voluntary compliance areas, here's free ammo. If you're going to be hunting on the Kaibab Plateau, where there's a lot of condors, we'll give you ammo to use. People are like eager to accept the ammo. We want you to remove your gut pile. People are eager to move the gut pile. That goodwill evaporates when it comes to mandatory.
Yep.
The goodwill evaporates when it comes to mandatory restrictions. And a lot of it's driven by some amount of efficacy for sure. A lot of that pushback, a lot of that goodwill evaporates also around cost.
Yeah. Well, I mean, it's hard.
Very different cost structures.
Hard right now.
To move away from lead ammo can be expensive.
Oh yeah, for sure. And I mean, just having ammunition shortages in general, trying to tell people to use non-lead when they can't find anything at all. So trying to make sure that people sort of know what their options are and don't just say, using lead is bad. Like, okay, if that's the only thing you can use, that's the only thing you can use. But, think about what you want to do after you pull the trigger.
Yeah. I think that it's keeping all that in mind is important, but I also think it's important that people realize you make choices, I make choices, right? And it is like lead kills raptors. Now, does that mean that we're going to lose raptors? That they're going to vanish from the landscape? Not necessarily, but it kills raptors.
Right. So it comes down what's your choice.
Yeah, it winds up being an uncomfortable reality that it's a thing that happens. And how often do they come through here that you find? And what is the symptomology?
So lead is really a tough thing that affects all sorts of different systems in the body. So it's a neurotoxin. It's bad for your kidneys. Like, it gets circulated in the blood and taken up into your bones. So you kind of lose it constantly through your bones. I had one case a long time ago with a mountain lion that had eaten a whole bunch of lead shot from something or other. And the lion was so rotten that we actually tested the bones in its paw, and it came out screamingly high for lead. So
it had been like metabolized
that fast that it killed it, and we could find it in its bones already. So it goes throughout the body. And I mean, the main thing that people focus on, especially like with children, is the neurological development. So it's not necessarily, at this point, if you lose a few brain cells from eating lead, it's, hard to tell the difference maybe. But for kids that are developing, that's the real challenge.
That work where they looked at rural people who had a high consumption of game meat versus urban people who had no consumption of game meat, but the urban people were having higher levels of lead
from paint chips.
Yeah, just environmental.
Yeah, we have had this working group on lead in New York for a few years. It's got Department of Health on it. It's got venison donation people on it. It's got Audubon to talk about all these different issues. And I've done work here with people in the communications department. And one of the things that we talk about is backlash. Like if you tell people like using lead is bad, then they're just going to dig in harder on that. Yeah. So that, really backfires in what you want to do. So just making people aware of what the issue is in a non-judgmental way, and letting them make the choice about what they want to do.
I remember sending in some garden soil one time, and the garden soil having
it's everywhere.
Super high lead levels. And the only thing you could think of is back from leaded gas.
Exactly. I mean, we used to have lead in everything. It's kind of interesting. That,
like leaded fuel would just would drift down.
Oh, yeah.
Right? And get in your soil. And then the recommendation was wash the produce. Huh. That it's like the plants aren't taking it up.
Mm-hmm. Yeah, it's just
you're ingesting it because it's folded within the leaves of your spinach, or it's like, leafy, like cabbage or whatever, things where it's hard to clean.
Yep.
We're more likely that you're going to be just consuming it, I don't, the lead thing, like one of the areas where I don't have personal concern, I guess because it's me or whatever, I never have personal concern about personally ingesting it. But it weighs on me about the wildlife issue. Mm-hmm.
Yeah.
Especially when I learned from you today about fishers.
Right. And it just depends. Some people are moved by the wildlife issues depending on what your ethos is. Some people are more concerned about it and, their own human consumption. It's variable, just the same way people hunt for a lot of different reasons, whatever is meaningful to them.
Crystal ball me on, give me some crystal ball stuff on CWD.
On CWD. Oh man, that's sort of tough. I
know researchers get real uncomfortable with the crystal balls.
I know, we don't do that. we're looking backwards most of the time. So
or just give me some thoughts. It doesn't need to be predictions. Just give me some, give me some thoughts.
Well, I'll talk about one of the things that we're doing that I hope people can get behind. So right now we have a large program called the Surveillance Optimization Project for Chronic Wasting Disease. And this started out as a way, based on what we did here in New York with doing more risk-based surveillance, because there's still a lot of states that haven't found CWD and they don't necessarily know where to look. Mm-hmm. So like New York had it in 2005, sampled really intensively until 2010 for their 5 years. And then they're sort of like, now what? Like, what do we do? So we set up this risk-based surveillance based on deer density, based on hazards, like, Pennsylvania's got a lot of CWD, so we want to make sure we sample intensively along the southern tier. And then going back to the, how do prions get into the state and how could they get spread? We want to sample more around those areas. And so we did this for New York, and then Tennessee asked us to do the same surveillance program for them. So we did that, and that was how they found CWD. And we've subsequently done it for 7 other states, and now Alabama found it, and then Florida just found it.
Okay.
So we said, okay, we don't want to do this just for individual states anymore. Like, states are struggling, spending a lot of money on CWD. How can they do their surveillance more efficiently and effectively? So right now we have... can I
interrupt you for a minute, though? If you did it for a bunch more states, doesn't it make sense that you'd have a bunch more states that would be that had CWD? Meaning...
Well, some of them already had CWD, like Pennsylvania and Virginia.
But Florida just got added to the list.
Yeah.
And they got added to the list because they went and looked for it. I mean, do you remember this conversation around COVID being like... There'd be these arguments that, well, yeah, there's a lot... You're seeing... High levels of COVID seem to go hand in hand with high levels of COVID testing.
But the thing is, it's not that they're not looking for it. It's just they don't know where. Like, they were doing surveillance, but it wasn't like necessarily emphasized in the right places.
I see.
So like Tennessee was doing about the same numbers as they were previously, but before they were getting samples where they were easy to get them. And so they kept coming from, the same places where we're like, no, look, in these 2 counties are your highest risk. And that's where they found it.
Yep, I got you.
So
So they might have been conducting, some number, 100 samples kind of pulled from roadkill deer.
Probably thousands of samples, but, just not in the places where they needed to be getting them from.
And so the idea or program or whatever that you were exporting was a more precise way to survey.
Collect data on where your risks are and then sample in those high risk areas.
I see. Okay.
And we also know from other states that have CWD, like adult males are more likely to have, test positive than, fawns usually don't test positive. So the test costs the same whether you test an adult male or a fawn. So put your money into that.
Yep.
We don't get a lot of adult males in just because they usually go to a taxidermist. So how can we engage with taxidermists to help us collect samples so that we can get those in and make it more efficient for the agency where the biologist isn't having to pull samples all the time, kick some money over to the taxidermist to have them do it for you. They're just going to throw that stuff away anyway. So, but every state sort of recreates the wheel in how they do this. And so they're collecting data separately. They're, spending a lot of time invested in surveillance and how they should do these things, when if we had a more regional or a more collective approach, we could compare data across states. Other states would know, like what their neighbors are doing, how much sampling they're doing, and they can have some decision-making tools associated with that. So the management agency can say, hey, we're mostly worried about, this particular area. What should we be sampling in there? How many should we sample? So we have software now that we're giving away to state agencies or tribes or provinces for free, that they can use this system and use it to whatever, if they want to upgrade from their Excel spreadsheet that they've been using, fine. If they want to run models in it to make decisions, they can do that. If they want to see what their neighbors are doing, it's useful for that. So that's sort of, as far as, maybe aspirational crystal ball, I really want to see wildlife agencies sort of come together with more standardized work that can be compared across the country rather than each state sort of doing their own thing.
And then if you look at a state, let's say take Florida, which was very recently added to the list of states that had a positive case in it. They get a positive case, then there's kind of there's a so what or not.
So what happens then, or what does that mean for the public? Or
meaning, let's say you're doing a bunch of testing. Let's say all the states are doing the same testing. You can't expect that all the states are going to have the same response to the testing.
No,
because there's some states and where the agencies have a perspective that's kind of like a little bit of inevitability. Doesn't matter. We're not going to do any real extreme containment measures. So that would lead you to be like, well, then perhaps testing isn't like valuable to them. If you're, so how are you, from a national perspective, how are people coping with that?
Yeah, I don't know.
The ambivalence or great care or whatever that a state takes about the disease, right?
I know. I sort of step out when it gets to management because it's so much people orientated in what they can do. So if a state finds CWD and they want to do something about it, their first detection is their best opportunity, because from a human-centric perspective, that's when people are fired up and care about it and like, yeah, let's get rid of it. I mean, I totally lean on New York, having been the only state to get rid of it, to be like, yeah, we're gonna try to kick its ass again if we find it, because that's the only real opportunity you have to do something. So it just depends on the management agency and what they choose. I mean, maybe like with Tennessee, their first detection, they had, over a dozen animals. So they're kind of like, crap, we're way further in it than, the very first detection. So like if you get a whole bunch all at once, you're like, okay, we've had it for a while and just haven't found this pocket. Now we know what to do with it. But I think you'd be hard pressed to find any agency that wouldn't do something, as far as like carcass or feeding or baiting, like bans that they would put in place on that to try not to to spread it further, because the thing that I talk about is, CWD pulls away resources from everything else in an agency. In some agencies, they have their fish biologists out there, pulling samples opening weekend of gun season. So it's, even if you don't care about deer hunting, like maybe your thing is butterflies, like it's pulling resources away. So everybody should kind of care about it at that point, because it's such a behemoth and it sucks so much money and time away from agencies that it's worth caring about. There's got to be some part of you, and I understand, if you got to punt this one, that's fine. There's got to be some part of you, because it definitely exists in my head, that it's just like an inevitability. Meaning, I know New York did it, but like you said, you think it's just going to be everywhere at some point? Oh man, like let's say I had to make a huge bet. Okay. Like every dollar I'll ever have or ever have had, and I had to bet it on that it'll. Meaning, this isn't a demonstration of what I'd like to see. Yes. But I just had to bet on what I thought would happen. Is there a time frame? I hate to say it, but I hate to say it, but I don't want to be dishonest. Like it just feels unstoppable. I agree with you to some extent, but my point is, like, what's the time frame on that? Are we gonna, young people right now feel that way about the earth, like that the earth is just gonna die at some point. But like, what's the time frame for that? So I feel like in a little bit of a way that's bad parenting. Okay. Well, we're not here to discuss parenting. Yeah, I don't know. It's one of those, like, fight the good fight. Like, do what you can while you've got it. I understand that, but I wasn't talking about that. I know. But I was talking, and I know it's counterproductive. It's counterproductive to point it out, but I just wish there were more. I wish there were more victories. Mm-hmm. I do too. And to me, and I've had friends of mine, and my friend Doug, I'm sure, is listening to every word I say right now. Hi, Doug. It pisses Doug off when I say this, but I've brought up to him, I've said, I used to say, if I somehow knew that it would never jump to humans, I would care a lot less. Meaning, like, it's clear what I mean. Yeah. Meaning, when I think about chronic wasting disease, I think about, it's never happened, but I think about how catastrophic it would be to deer hunters. Oh, yeah. To deer hunting, if we suddenly had these cases, in some community or some family, that they develop some novel, version of mad cow disease, and I think it would Jakob Creutzfeldt, whatever the hell they call it, and it killed some family, and it turned out that family had eaten some quantity of infected deer meat, and it became, the holy cow, right? Oh, yeah. It'd be catastrophic. It would change the way people think about deer. Mm-hmm. It would change the way people... cockroaches, pigeons, rats. It'd be like, you look at deer and you get excited. It's great. People are excited to see deer. They're excited to see deer tracks in new places. Like, and if it was a disease causing animal, it's so devastating, right? And you can look and be that. And I remember one day you and I had an argument about this, where I said, and I stand by it, I've said hundreds of thousands of people have eaten CWD infected deer. Maybe it was you, maybe it was someone else that said hundreds, not tens. And then that caused me to think about it a bunch. And I thought about it a bunch, and man, you're sticking with hundreds? Hundreds. Okay. Hundreds of thousands of people have eaten CWD infected deer meat. Like, it has to be true. Yeah, I don't think so, but okay. Sure. How about 99? 99 people? 99,000. No. Come on. I don't think so. Okay, give me your number. Lower. All right. Tens of thousands. Price is Right. Tens of thousands. There's no way you're going to argue.
There's a case for 100 people in one meal.
I know. That was in New York. Yeah.
Okay.
I'll give you 10,000.
Okay. Hundreds of thousands. Tens of, okay, whatever the hell it is. A lot.
Okay. People have eaten infected deer and have not gotten CWD that we know about.
That we know about have not yet. Okay. So I look at that and that's heartening. I'd rather that than the opposite.
For sure.
Anybody would. But people, there's no one that can come and tell you. There's no one that can come and tell you. And if someone does, like you can't believe them. There's no one can come and tell you that it's impossible.
Right.
So in the past, I have brought up that if I knew somehow as some fact, which is impossible, but if I knew as a fact that it wasn't possible, my concern about it would go way, way down. But now there's emerging evidence in places where they're seeing evidence of population level impacts. Because it used to be that you could look and you'd look at these areas of CWD and they oddly would be areas that had so many damn deer.
Mm-hmm. Right. Yeah, because high density.
Like
and
there's
then
CWD in southwest Wisconsin. It's like, why does southwest Wisconsin have so many deer and they got all these giant bucks?
Mm-hmm.
So how do I look at that and get worried? If I'm not worried about catching it and dying, and there are places that have a lot of deer and they're still shooting big bucks, I'm not going to worry about that. But this emerging stuff, and I haven't looked at it a lot, but I've heard it, I've seen it demonstrated by people that I have faith in, that it's beginning to see where the populations can't support it.
Exactly.
And that you might wind up down the road with really damaged deer populations.
Exactly. I mean, we were talking earlier about kids and hunting. And so when I first started studying CWD 20 years ago, I'd say, this probably isn't going to impact you. It's probably not going to impact your kids, but it might impact your grandkids. And now the speed with which this, like the prevalence curve, the exponential curve of how the disease takes off once it gets hold, like I'm worried about my son hunting and like the opportunities that he's going to have, or if he even wants to, because, it's not much fun going out and, shooting a deer and you come up to it and it's all scrawny and sickly. And I mean, like on Doug's place, they can practically identify them by sight now because they know they have the disease and like, yeah, that one's probably infected.
Oh yeah.
So,
when you're shooting deer in an area and the majority of the deer you're shooting are positive.
Yeah.
If you've made the call that you don't want to eat it. I came up with a test. I haven't done it yet.
Okay. I'm curious.
When I'm talking to friends of mine, and I got a lot of friends who really they're not hostile. Like they'll listen to us have this conversation. They're not hostile to discussing it, but in the end of the day, they don't care.
Yeah.
And it's like these are not dumb people. It's like they don't care because they have a hard time with the population level decline. Especially when people say, hey, the cure to CWD is to lower deer numbers, or else they might get a disease that will lower deer numbers. And then it's like, okay, well, is the medicine worse than the sickness? Mm-hmm. You're telling we should kill all of our deer so that all of our deer don't die? It's like a challenging logic.
Yeah.
I'm just trying to give voice.
Back to the burning down the village to save it.
Yeah. Burning the village to save it. And also, they're like, no one's gotten it. What are the chances I'll get it? And so really smart people who are not adverse to scientific information have decided that they're not concerned. Okay. Or they're not as concerned as they are about other stuff. And I'll debate this with them, but I formulate, like, there's a test I would like to do. And I've talked about this before. I would like to go and get deer meat. I'd like to find 20 CWD positive deer. Sick looking ones, and get all that meat and make a burger. Okay. And I make a big batch of burgers. And when I meet a friend that says they don't care,
feed them the burger.
I make them that burger. Mm-hmm. I fry them that burger.
Yeah.
Okay. This is me making the patty.
Okay. Good visual.
I fry it just how they like it.
Mm-hmm. Medium rare.
Yep.
Because prions aren't killed by cooking.
Medium rare.
Right.
Pickles. Okay.
Oh, pickles.
And I would say to them, I'm like, man, the minute you eat that burger, I'll talk to you about how you don't give a shit about CWD.
Exactly.
And I want to know how many burger takers. Now I've got people that said, you do it, I'll eat that burger.
Okay.
But man, here's the thing. If I made that burger and someone said, okay, I want you to give those burgers to your kids now. Dude.
Right.
Sitting where I'm sitting right now, there's no, even if I could hide it from my wife.
Which is probably not smart.
There is no, I just like right now, what I know right now, and I like pay a lot of attention and I try not to be alarmist, right? And I'm not alarmist. I could not give that burger to my kid.
Yeah.
I couldn't do it.
I agree. I couldn't do it either, knowing that similar disease in sheep, scrapie, they've never had a documented human case that they've known about. Even with mad cow, there, I would agree with you about the hundreds of thousands of people. It was only, there's been 200 plus people that have ever gotten mad cow, and they likely have some genetic makeup that, made them susceptible to getting that. And I suspect it's the same way that there is some genetic makeup in humans that would allow that prion to cross that species barrier. And
see, now you're making me want to eat the burger.
But how do you know, right? Like, so I don't.
Can they? Yeah, well, that's what we need to find out so you can test, and they give you the green light on burgers.
But it's very human centric, isn't it? Like, we only care if it's going to affect us. Like, why not care if it affects the wildlife population? Why not care that deer are dying this horrible disease that puts holes in their brain? We say we love these animals, but yet we're not willing to do the few steps or let the management agency take some action in the short term to try to stop that disease because it might be inconveniencing us.
I'll tell you a thing. Let's move on from burgers for a minute. Let's just talk about big giant bucks.
But now you're making me hungry.
I know, but let's talk about big giant bucks.
Okay.
This is to my friends who aren't concerned about eating burgers. They haven't yet seen or are convinced about, they haven't yet seen and are not convinced about population level declines, but they love big giant bucks. And all of their recreational free time is put into the finding of the making suitable habitat for big giant bucks. Now, I was recently in a room with you and we were looking at prevalence rates and the fact that it's always fatal.
Right.
Always kills you.
Yep.
There's no getting around it. You get it, a deer gets it, that deer's going to die.
Yes. Probably sooner than it would have otherwise.
Takes 6 or 7 years to make a trophy buck. I mean, sometimes less, but like, right.
Yeah.
I mean, you can have a trophy buck at 4 years, but it takes a long time to make a trophy buck. When the disease becomes so prevalent that a deer's not going to get to 2 years of age without getting it.
Right.
Because prevalency rates are like, prevalency rates among 2-year-olds get to like 60%, 70%. It's always fatal. It's fatal within a couple years. The idea that you might be that if that ideal big buck is 5 years old, 6 years old, whatever.
Yeah, you're not going to get there.
That you get to be where you're still seeing tons of deer on the ground because there's a carrying capacity to the landscape, right?
Right.
You might still be seeing, there's like, yeah, you go out and see deer all the time. But you just start to be that, man, I don't know what's going on, but they are not getting to 5 years old.
Nope.
They're not getting to 6 years old because they're getting sick at 1 or 2. And they're succumbing to the illness at 3 or 4.
Yep. So
and they just aren't making it through because so few make it through anyway.
Right.
Remember talking earlier about additive mortality?
Yep.
So few make it through anyway. That they don't get shot by someone. They don't get hit by a car. They don't get killed by a coyote. They don't get hung up in a fence. All the shit that kills deer.
Yeah.
That all of a sudden there's this other little thing that just kind of helps guarantee they never get that old. That will make people.
Yeah. I mean,
it's like, that's perhaps we're already seeing that. And one of the things that I think about when I think about CWD is I wish that all areas had a lot more tracking of not just Boone and Crockett, but like a lot more scoring trafficking. It'd be very interesting if you could have gone back and looked at counties, how many Boone and Crockett entries were counties getting,
Right.
and do you see Boone and Crockett entry
as much anymore.
lowering
Yeah.
in areas that have had long-term infection? Because if what it seems to be true is true, I think that you're going to have yet another thing.
We'll steal that idea for
prevents
Corinne's
bucks.
master's project.
Oh, when Corinne goes back to college?
Yeah.
That prevent another thing making bucks not be 5 years old or 6 years old or whatever the hell.
Right.
Trophy bucks. That, man, I got friends that'll go out and spend days hinge cutting trees and planting this and that, right?
Mm-hmm.
And if you told them, like, man, you know what your biggest problem is about making big bucks and getting big bucks is they're not going to live that long anyway, dude, because of a disease. That
would change their tune.
Yeah. Me, it's the eating.
I know. Well, I mean, people, you're called MeatEater for a reason, right? Like, people hunt for different reasons.
Yeah, that's not why I gave it that kid. That's not why I came up with that name. You know why I came up with that name for the show?
No.
I had a little teeny kid, and we'd read a lot of books about animals, and we'd read books about dinosaurs, and you get to the T-Rex chapter. This ferocious meat eater. That sounds a cool word.
Very good.
I like the word saber-tooth, too.
Oh, yeah.
It could have just been that all along.
Yeah, there you
But
go.
either way, I eat a lot of deer meat.
Yeah, me too.
And I get and that scares me, man.
Well, that's the thing. Everybody's going to have a different, just like the lead, like people are going to have different reasons for caring about something. So you asked about ecology. Like, I try to give people a suite of like all the different why they should care about these. Like, maybe they don't care about deer populations. Maybe they just care about what it's going to do to them. Maybe they care about big bucks. Like, try to give them enough information that they can make an informed choice about it. But
you mentioned a thing about eagles and lead. And I think it's true of deer diseases. There's another thing I think about when I sit around worrying about CWD is like, there's no doubt that human activities are increasing the spread.
yes, I agree.
It's not really a debatable point. I mean, there's other things spreading it, but humans are facilitating increasing spread. We are stewards of the landscape.
Ideally.
Okay? Ideally, we're stewards of the landscape. Do you, if you love deer and you love deer hunting, is it in you to be like really okay with a disease that kills deer?
Yeah. That's why I don't understand how people can be like, no big thing.
Because someone's going to, I want to talk about this before we close. Someone, and I do it too, is going to be like, well, what about EHD?
Yeah. Well, we can talk about EHD.
Okay. What about EHD? I mean, here's a disease that knocks them hard. It's got to be painful.
Sure. But they...
Yeah, give me the butt.
Okay.
So I say that. We're talking about CWD, and I say to you, yeah, well, what about EHD? That kills all kinds of deer.
So EHD, epizootic hemorrhagic disease, and we'll throw in blue tongue, which is another hemorrhagic disease, and just call them HD for hemorrhagic diseases because they function pretty much the same.
So I said, okay, what about HD? That kills all kinds of deer.
There you go. Yes, it does kill deer, but not all deer. It's not fatal like CWD. And in New York, we've been doing another study on this because we had some pretty significant outbreaks in like 2000, which was super inconvenient timing since, the pandemic was going on and people were in their houses and just kind of trying to deal with a big deer die-off. So it started in the Hudson Valley. And we started tracking reports from the public. The biologists were going out and collecting deer. It's challenging to...
Can we include the creepy part?
Sure. Which one?
Floating in ponds.
Yeah. So, okay.
Laying next to the creek, floating in ponds, in a water trough.
So with hemorrhagic diseases, the deer get a bad viral infection, and so they get very feverish. So a lot of times they'll go to water, which is why you find these deer around water. But we have to be careful because we mentioned that we had a rabies positive deer a couple weeks ago. Rabies, a lot of times we'll find the deer in water too.
Okay.
Again, another viral disease. They tend to go to water when they get feverish.
We got to do a big digression. I'm sorry.
Okay.
Because this is the thing I've always wondered about. How can rabies at one time be that they're drawn to water, but then also in humans it induces hydrophobia?
Yeah. Well, I mean, you can be thirsty and go to water, but then the paralysis of being able to drink. So it might not be that you're afraid of water. It might be that your body stops functioning to allow you to drink.
But I thought so it's not that people get scared of water.
Yeah, I'm not up on the human.
Well, they used to call it hydrophobia.
I know, but yeah, it's more that they're not physiologically able to drink anymore. So they're very thirsty, they're feverish, but they can't drink.
Do you know I defended my family from a rabid squirrel one time?
No, but please tell.
It was in this state. We were visiting a friend.
Uh-huh.
A squirrel came my kids were all swimming in the pool. The squirrel ran up to the edge of the pool, very agitated, started running up and down the pool. Like if this is the pool, my kids are like sprinkled throughout here. And the squirrel's going like this, back and forth, back and forth, back and forth, jumps in the pool, starts chasing kids around.
No.
I grabbed the squirrel by the tail and gave it a...
Yeah. Squirrel flinger.
Yeah.
Good job.
Had to have been rabid.
Rodents, we don't see a lot of rabies in squirrels. Rodents don't get it as often, but we just did have the beaver that was positive for rabies.
Well, how else would you explain what that squirrel was doing?
Squirrels have a lot of issues. I don't know if you've noticed this.
I don't know.
Yeah.
You guys just had a rabid beaver.
Yeah.
He bit 5 people.
He did.
You make a movie about that.
There you go. You're the production company.
Okay, so back to EHD.
Back to EHD.
So deer could get rabies.
Yes.
And they'll wind up in the water.
Sometimes, yeah. But if I hear about deer in water, I usually think rabies or hemorrhagic disease.
Okay.
So why should we worry more about CWD than hemorrhagic diseases? Even though we had a big die-off, there were, over 2,000 dead deer reported. We don't know how many actually died in the area that was affected.
In a short period of time, huh?
Yeah, one summer.
Wow.
And then the next year, we also had pretty significant die-off, but it was more sprinkled around the state. So Hudson Valley, up towards Lake Ontario, and then on Long Island. But what we did in those hunting seasons was we collected spleen and we collected blood from hunters, and we looked for antibodies to the viruses to see if any of the deer survived. And we found a pretty good proportion of deer had been infected and then survived. So those are very sort of short-term transitory mortality events. And then last year there was nothing, no big mortality. So it might hit local areas pretty hard and knock the deer down because that virus is transmitted by a biting midge, helicoides, and those don't travel very far. So we're still trying to figure out the right environmental conditions, whether they like it dry or they wet, for breeding in mud flats. And the deer seem to rebound really quickly with those. So that is something that they can build up immunity to and, continue on without a long-term population impact. It might be short-term, but, over the long term, it's not going to, do as much damage as chronic wasting disease.
And I'll point out, too, that you don't have to have that nagging fear about catching a brain disease.
That's true. Yeah, we generally recommend people don't eat stuff they find dead. So hemorrhagic disease, they kind of turn mushy real fast because it's usually in the heat of the summer.
Got it. Well, I don't mean that about finding dead deer and eating them.
Yeah,
I just mean
the concern about disease.
Why eat, as much as I was playing devil's advocate to point out why not worry about EHD, because that kills a bunch of deer. And I would just say, added to the fact that there's some resiliency, that they bounce back quickly, that you can get it passed through a deer herd, and then a couple years later it's not in the deer herd.
Right.
Is different. But then there's also that later when you harvest a deer that had it,
you're not
at.
Right.
Well, we don't know if you're at risk. You're not at potential risk.
There you go. Look at.
Do you I don't know how well you can word this. This is my last question about CWD. Do you don't eat it? I'm just asking you personally.
Would I eat a CWD positive deer?
Yeah.
No.
Why not?
I don't have to. I have a choice there, so I'm not starving. Like,
okay.
I think the risk to humans is very small, but why chance it?
Yeah.
I mean, honestly, with the field work I've done and taking CWD samples from live deer, I've been in their mouth. Like, I was taking tonsillar biopsies when I was doing my research. So, like, I've had CWD positive deer breathing in my face that I know they were positive. So yeah, I'm probably going to donate my brain to science, like when I kick the bucket, just so people can check for prions.
We've been talking a lot about, we're trying to get someone from the Body Farm. Where's that place?
Tennessee, I believe.
Yeah.
They don't, if you want to donate your body to science, we heard that they're like,
are they not taking more?
Everybody wants their body there.
Oh, really? It's a popular thing now?
Yeah. They can't accept them all.
Well,
we heard
maybe
from
you
somebody,
can get on a wait list.
We heard from somebody that did some work there. Do you know raccoons, like, when it comes to carcass preference on raccoons, they like people who have diabetes?
Interesting.
Yeah.
I did not know that.
Do you know that?
I mean, just from this person who wrote in.
Yeah.
Yeah, I would verify that. I mean, I didn't know raccoons were that picky about
they like thighs and buttocks.
Well, don't
Makes
you?
sense. Yeah.
I mean,
for sure.
Okay.
Diabetes.
Interesting. I'm going to have to check this one out.
We're going to try to do a site visit down there.
Okay. That'll be good. Now that you got some more dead animal, can I come with you?
With where you hang out and work, you'd probably have zero problem with that.
I don't know. Humans gross me out a little bit. Like animals I'm okay with, but people are kind of gross.
I was, when we did our tour today, I was thinking that, I was thinking about the resiliency of your colleagues.
The ones that have to cut up the dead stuff?
It's a lot.
Oh.
Smells.
It could be, it's a lot. It's like, just the environment, right? That lab environment and just the what's coming through the door, right? It's a lot.
Yeah. I mean, but
I'm sure you get used to it pretty quick.
Right. Yeah. It's just what their job is. I mean, you think about people working, like nurses having to deal with live people complaining all the time. Like, at least these are dead. So,
well, there's a difference though, because I don't get that it's a lot feeling hanging out in a butcher shop or in a slaughterhouse. That doesn't, because it's like food production. But the disease element, the sickness element.
But see, they find joy in that.
They do.
So
he's talking about people that really like kidneys.
Exactly. They get very excited. I mean, you mentioned nasal bots previously, and when I was doing the helicopter capture for my PhD to catch deer, we had a mortality, and so we euthanized it. And one of my fellow grad students who loves dead stuff, started doing the necropsy, and it was chock full of nasal bots. And
where was this again?
This was in South Dakota.
Okay. Yeah.
And she was so excited. I mean, she had 2 handfuls full of nasal bots and came like skipping around to show us all, and just thought it was like the best thing ever.
We had some guys in Sonora one time tell us, some cowboys in Sonora tell us that thing does the thinking for the deer.
Oh, interesting.
It'll take over and do the deer's thinking.
Huh.
And the deer does its bidding.
All right. You were asking about ticks earlier at the CWE conference in Denver recently. They found prions and nasal bots too.
Oh.
Yep.
Sure.
So.
And the fact that one day he just goes, huh, It's a hacks.
I know, a big old nasal
hacks
bot.
that bug out and it flies
know.
away. That'd be a good video to capture.
There you go. Aspirational.
So how do people? What's the best when you want to wake up in the morning and find out about wildlife diseases? What do you like to read?
What do I like to read?
Yeah, like if someone wants to track wildlife disease issues, let's say someone's so inspired.
So
by your work.
I am
your line of work
with the Cornell Wildlife Health Lab, which is part of the New York State Wildlife Health Program. You can look up Cornell Wildlife Health Lab. We've got lots of fact sheets and information about all the research that we do. You can go, New York State has a great website. There's other states out there that have really good information depending on, where they are. It's probably more applicable to them. So check out your state agency website and see what wildlife health information. But if you want general stuff, you can always come to Cornell Wildlife Health Lab.
And don't try to bring a dead animal down here.
Well, you can. Just call us first.
Call us first, yeah.
Yeah.
You can't
We're happy
show up.
Well, you can, but it's
just bang
less
on that
pleasant.
big door out there.
It might take a little bit. I might get a nasty phone call when they're like, "Who is this person?"
You'll know you're at the right door.
Yeah.
The door is so tall. The intake door.
Yes.
Is so tall.
You can back a semi in.
That it is known to have accommodated elephants and giraffes.
Yep.
Look for that door.
That's the door you want.
If you say to yourself, "That's not going to accommodate an elephant," you're at the wrong door.
Can't put an elephant through there.
Well, thank you very much for coming on the show.
Absolutely.
It's fascinating work. I love wildlife, and and it's good to know that there's people out there monitoring wildlife diseases. And it becomes upsetting to me when there's things that there's preventable things we're doing.
Yeah.
That could make it that less wildlife vanishes from the landscape because of diseases. So it's good to have a heads up.
Yeah, I think remembering we're part of the ecosystem, and if we can be part of the solution rather than part of the problem, that's always a good thing.
We're part of the disease ecology.
We are.
Yeah. All right. Well, thank you for coming on. Appreciate it. Krysten Schuler. Thank you.
Thank you.